So the biopsy collection is made up of a few steps. At first, we have to prepare the cantilever itself to be able to receive the biopsy. And for that, we apply a vapor deposited coating that makes sure that the RNA that is being collected is not going to absorb on the microchannel surface. And that's very important to make sure that every RNA molecule is kept safe in the biopsy. And so we just use simple vapor deposition coating for that. That can be reused. And then we just have to fill the cantilever with oil. It's a sterile mineral oil to create an interface between the biopsy and whatever else is in the microchannel. And once that happens, then we can optionally preload the cantilever with a buffer. And this buffer solution can already contain some RNA inhibitors. So again, since every RNA molecule is precious, we can make sure that the RNA's enzymes are not degrading our sample. And that's basically the preparation part. So that's when the cantilever is ready to actually interact with the cells. And so at that point, we can go into the sample that contains the cells and then simply target which cell we want to take the biopsy from. So this works in a point and click way. We just click on the cell that needs to be sampled. And then the cantilever is lowered into a cell with a force control mechanism. We pierce the cell membrane, and then with negative pressure, we take out the biopsy. And as I mentioned before, this is happening through a live volume measurement as well. So we can always monitor what is the volume that is being taken. And once we reached this desired volume, which is actually usually 1 picoliter, can be a little bit more if the cells are bigger. Once we have this, then we stop. So once we have enough volume in the microchannel, then we have to stop the pressure and then simply retract the cantilever from the cell. And once that happens, we have the biopsy secured in the channel. And then, of course, we have to deposit it for downstream analysis. And since this is only 1 picoliter roughly, of course, if we deposit it in air, then it would evaporate immediately, and that's not very useful for RNA analysis, for example. So what we do instead is to deposit this 1 picoliter biopsy into a droplet, and this is a much bigger droplet, so 1 microliter. And that's really the end of the process, that we have a 1 microliter buffer droplet that contains the 1 picoliter of biopsy. And this is how we can interface the FluidFM collection, biopsy collection, with the downstream analysis, because then this 1 microliter droplet that contains the RNA or whatever we are trying to analyze, this can be then prepared for the next step, the first step of the downstream analysis.